afternoon, everyone. I think it's about time. So welcome everyone to the 32nd Special CU Asahi Glass Foundation seminar. The theme for this seminar is Research and Innovations for a Sustainable Future, Bridging Technology, Environment, and Society. My name is Tirapong Buwusha. I'm from the Faculty of Science, and today this is the first session, which is under the theme of life science. So today we have a great lineup of 7 speakers, and for each speaker you have 50 minutes and 10 minutes for presentation and 5 minutes for questions. So let's get started. The first speaker is Associate Professor Pirapon Vetikul. He's from the Department of Computer Engineering, Faculty of Engineering. He will give a presentation on the automatic gastric intestinal metaplasia segmentation system from gastroscopic images. Please join me to welcome Ajahn Pirapon.
Thank you very much, Krab. Please allow me to introduce myself. I'm Professor Pirapon Vetikul from Department of Computer Engineering. First of all, I would like to thank to Asahi Glass Foundation to support this research funding. This research is part of our product called Deep GI, which is AI for GI tract though. I have to say that one of the problems for GI tract is colorectal cancer. That is the third most common cancer in Thailand. You know what? Last year, we have 16,000 new cases or 44 new cases a day. And 1/3 from this number, they pass away. That's pretty high rate though. So this means that we have 15 people who dead from this kind of cancer a day. Anyway, this kind of cancer can be prevented if we have early detection, such as in US that the death rate has been dropped for the past 10 years due to effective pre-screening process though. So that's why we create a collaboration between our Faculty of Engineering and Medical School from Ajaran team and also from a company called ESM Solution to create an innovation called Deep GI, which is AI solution that detect anomaly in GI tracts. As for Deep GI, it is this box though, and it can render to show anomaly in real time. It's compatible with any endoscopic camera, Fuji, Olympus, Pentax. And lastly, we extend our solution to compatible with many organs in GI tract, such as here, colon, to detect colorectal cancer, stomach to detect gastric cancer, bile duct, yes, is all of these are in progress though, yes, to detect cancer in bile duct though, bile duct area, and this solution can deploy in real environment atฬาลงกรณ์ Hospital. So as for the funding that we get from Asahi Glass Foundation, we use that fund to improve our model to detect cancer in stomach much more accurate though. I have to say that for gastric cancer, it's the 5th most common cancer, and especially for Japanese people, Chinese people, Korean people. This kind of cancer is even one of the top ranked cancer that they have to face though. So pre-screening is very, very important. So that's why we have a protocol called EGD protocol that they put something called gastroscopy from the mouth, yes, to check the stomach area. And to check the stomach area over here, we have to detect the lesion that has some problem called GIM, something like this. And for the gastroscopy camera over here, it has to support white light mode and NBI mode, something like that. So this is some example. As you can see, this in white light mode though. And it detects the lesion that has some problem called GIM, gastrointestinal metaplasia. Yes, is one of the precancerous state for gastric cancer. Yes, try to detect something like this. This is still in white light mode though, still in white light mode. Once the doctor, they identify some incident or something that very suspicious area, they're gonna turn the color mode into what we call NBI mode, narrow band imaging. So they can see the contrast and they can see the texture much clearer. Yes, these some example. That's pretty scary though. So this kind of cancer, if we can pre-screen in early stage though, it can be cured. Yes, the problem is from our preliminary work, we found that the model, we cannot use universal model because there are multiple color mode, white light mode and NBI mode. That, as you can see that we need to see the texture though. And once we see the NBI mode, as you can see that the problem is it's false positive. Too much detect error. That is not accurate at all for positive rate, very high for positive rate. And especially when we close up the camera. So every time we close up the camera and it NBI mode, even though it is a normal area, but it tend to predict as GIM area or anomaly area, which causing very high false positive rate. So that's why we need to improve our model though. And lastly, again, since texture is very important features, doctor normally notice from texture. So if we can increase the image resolution, so we can see the texture much better. So that's why in this work, we propose a new model. We run on a very high spec of computer with many variation. So we collect new data, yes, to train data with more variety of normal case and support with multiple colors. We design new model that can perform in real time. And as you can see in our new model, there are two parts though. First part is for white light mode. Second part is for NBI mode. So it can be specialized in each color mode. And it also have color selector module to decide that at the moment it's white light mode or NBI mode though. And we also design many variation of the model. And lastly, we also try to increase the image resolution. But the drawback from this one, the inference speed can be dropped a lot. So that's why we increase the image size to fit to the model, while we try to compress the model so we can compensate the speed over here. And we proud to show that from our previous model, the baseline model and the new model, as you can see in the white light mode, it's improved, but much improved in NBI mode that we have very high false positive rate. But right now, as you can see, the accuracy reached 97% and overall performance is reached at 92%. That's very high though. And there is some video result over here. Yes. So our model right now is specialized and support both white light and NBI mode because we have two parts in the model. This white light mode. And this is the NBI mode, the one that has a lot of issue with false positive rate though. But right now it's much better. We can reduce high number of false positive rates based on our new model that we get scholarship from Asahi Glass Foundation over here. So as for outcome of this work, we just publish a tier 1 journal. Yes, in the journal called Knowledge Based System this year. Yes, this is the outcome of this work though. I just want to say that since we have DCI as our data product, so we already integrate new model into our AI box, DCI box, and we has this box has been using in many hospitals with a lot of patients. But as for new model that we got scholarship, that we got funding from Asahi Glass Foundation over here, this one of the thing that we are so proud that yes. We, the doctor team, do the operation and broadcast it to Korea. Yes, in the conference in real time from the operation room. So that pretty much it for our outcome. Yes, that we get a new model. We publish one journal in tier 1 and we also deploy it in the real hardware. Thank you very much. Thank you.
Thank you, Ajahn Pirapon. So now the floor is open for questions and comments.
Thank you very much for your presentation. Can you hear me?
Yes.
Okay, thank you. It's great technology. I think the real-time diagnostic is very important for this application. So one of the key is speed for the image calculation or something like that. If you improve your CPU or GPU improved, your accuracy or predictivity will be improved much more.
Thank you very much for your question though. At the moment, since with our solution, we concerned about real-time inference though. So the hardware that we use right now, the GPU, the version 1, we already test it and ensure that the GPU that we use is enough to run our model. So to answer to your question, if we increase GPU, can our model get better result? It can be possible if we increase the model size, but with this size of model, with 92% accuracy at the moment, yes, the GPU that we deploy right now is enough though. But in the future, it's ongoing in the future. If we increase the GPU size, the model can be bigger and it can improve accuracy as well. Yes. Thank you very much for your comment.
Thank you.
Okay, thank you for your presentation. I'm interested in the lighting or the noises for the photo and the images. How does the segmentation, the algorithm, handle the variation in the image the quality, image quality such as the lighting or the imaging noise? Please let me know.
Oh, of course, that's a good question. First of all, I have to say that we have a strong collaboration with the doctor team. So first one to handle the noise and variation of image though. As you can see over here, we collaborate with doctor team and we collect many variation of data set though. So we have case one, yes, as you can see, we have training data for white light mode, training data for NBI mode, so support both color mode, and also support many variation as well. So first one to answer to your question, we prepare a lot of very high quality of data that cover a lot of variation. Second one, to train the model, we have technique called data augmentation. So we incorporate with many data augmentation technique, flip, translate, rotate to increase the variation and the data size as well. So the model can tolerate to the noise. And lastly, in terms of model, we pick the best model, best real-time model that can cover up that part as well. So due to high quality of data, training technique, data augmentation, and in terms of model as well, that's why we get very high accurate model. Yes.
Oh, thank you for your presentation, and I can understand very well, and it is very important the photo and the image, the quality. Thank you so much.
Thank you very much.
Any other questions or comments?
Final remark though, this solution is deployed in real environment. So, and we're still open for more collaboration in the future. If you're interested in our solution, please feel free to contact me and our team as well. Thank you very much.
Ajahn Pirapon, one more question, please.
Last question. Thank you, Ajahn Pirapon, for the presentations. I actually have, I think, I'm not familiar with the field that you're studying, but I'm wondering if, different doctors actually perform this, using, I don't know, different cameras or whatever. Would that kind of affect the model that you generate at all?
Okay. Thank you very much for your question as well. At the moment though, yes, since the domain is pretty niche though, it's very small area. So the version of camera that we tested at the moment, we already test in real environment. So the camera that we use over here, we use what we call gastroscopy, and it can be from Fuji, from Olympus, and from Pentax. So due to that, we tested our model for a very long time. So we have image from all the brand and from all many color mode and many version as well of gastroscopy camera. So to answer your question, we already take that into account and train the model to cover almost all the cases. Yes. So if the doctor use different brand or different versions, different model, it can still work in real. Right now, to be honest, we are in the clinical trial stage though. Yes.
Thank you so much.
Okay, thank you so much, Ajahn Pirapon. Okay. Next, we shift gear a little bit to natural science. Our second speaker is Assistant Professor Dr. Nat Songwarawit. His presentation will be on analysis of metal element deposition in the cuticle of beetle weapons. Please join me to welcome Ajahn Nat.
Hello, thank you very much. Good afternoon, everyone. I am Nat Songwarawit, and the topic that I'm going to talk is about elemental deposition in the cuticle of beetle weapons. Okay. Oh, sorry. Okay, I would like to begin with the general knowledge about insect cuticle first. Insects are the most diverse organism in the world, and the body are covered by cuticle, nearly entire body. And insect cuticle act as an exoskeleton to support the body as well. Cuticle or insect may be hard and rigid, that act like an armor to protect insect from predator, or it may be soft and flexible, as we can find in body wall of caterpillar and abdomen of many insects. Insect cuticle can be divided into 3 layers: epicuticle, exocuticle, and endocuticle. The main components of insect cuticle are proteins, chitins, and small amount of hydrocarbons. And the flexibility, the softness, or the hardness of insect cuticle depend on the proportion of each layer of the cuticle and also the cross-link in the polysaccharide chain in the cuticle as well. And based on the chemical composition of the insect cuticle, it means that the basic element that should be found in insect cuticle must be carbon, hydrogen, oxygen, nitrogen, and sulfur. Anyway, there are some study reported that not only the basic element that I mentioned earlier, but also trace element and metals can be found in insect cuticle as well, especially zinc and manganese that are usually found in the area around the cutting edge of the mandible, especially for the insect that prefer to feed on food with tough or hard texture. For example, leaf cutter ant that feed on leaves, termite that feed on wood, and some beetle species that feed on grains and seeds. And scientists believe that zinc and manganese can increase the hardness of the cuticle and also improve the mechanical strength for the mandible of this insect. Anyway, most studies usually focus on the mandible of insect that used as an organ for eating food. But in reality, not all insects that use their mandible for eating. There are some species that use their mandible for other purposes, such as use as a weapon for fight or protect themselves from predator. So this leads to my research question. What about cuticle on mandible that are not used for eating food? So in my study, I selected stag beetle as the model for this study. Stag beetles are insects in the group of beetles. There are approximately about 1,500 species around the world. And the distinct characteristic of stag beetle can be found in males. Males have large and long mandible that used as a weapon for fight, while females have smaller body size with short mandible. And for male stag beetle, they don't use their mandible for eating at all, but they use their mandible as a weapon for fight with other males to compete for females. So, sorry. The objective of my study is to investigate the disposition of metal and other elements in the cuticle of weapon parts or mandible of stag beetle. And here is my workflow in my study. I began the study by collected insect specimen from their natural habitat in northern Thailand. I use light trap to attract them because stag beetle are active during the night, so I can use the light to attract them, come to the trap, and then I catch them. And here are stag beetle that I got during the field trip. Unfortunately, most stag beetle species that I got with a few numbers, so it is not sufficient to conduct the statistical analysis, except just only one species that is common red stag beetle right here. The scientific name is Prosopocoilus astacoides, and from this slide you can see the difference in the body size and morphology, but it's just because of the morphological variation in the population. The rightmost one is female, and the other three individuals are males. Males of this species appear in three distinct forms, based on their body size and mandible shape. The biggest one is alpha male, and the medium size is beta male, and the smallest one is gamma male. Firstly, I would like to examine the position on mandible that is important for fight and frequently contact the opponent during the fight. Because I believe that not all region on the mandible that is important for the fighting success, and there may be some regions on the mandible that might frequently contact the opponent. So it means that this region need hardness and mechanical strength more than other region. I conduct the experiment by conducting the behavioral observation. Please take a look at this video clip. From the fighting behavior, you can see that the region that frequently contact the opponent is around here at the tip of the teeth, both at the apex of mandible and also at the base of the mandible. And for the fighting style of stab battle, they usually try to use their mandible, grab the opponent, and try to lift the opponent above from the ground or above from the subject that they are clinging on. And the fight will be over when one of them retreats from the fight or being thrown away from that area. Okay, and the next step, I utilized stab battle and conducted dissection to separate mandible from the body. And then I analyzed elemental composition on the mandible by using SEM EDS or scanning electron microscope coupled with energy dispersive X-ray spectrometer by using point analysis. And based on the fighting behavior of stab battle, I decided to examine the elemental composition from three regions of the mandible. The first region is the tip of the teeth, because this area is the area that frequently contact the opponent during the fight. And the next is intermediate region or edge between teeth, as indicated by blue arrow. And the last one is median area of the mandible, which is the part that contact the opponent with the lowest frequency, as indicated by yellow circles. And here is the result from my analysis. Besides basic element that I mentioned earlier, I found trace element and metals for 9 elements in total, as shown in this table. And of these, there are 3 metals, 3 elements that I found in the cuticle of stab battle, including titanium, nickel, and aluminum. And by comparing between regions on the mandible, you can see that the tips of the teeth contains all of these 9 elements. And these pictures show the quantity of trace metal and trace element and metal found in each region of the mandible. For the overall picture, you can see that the tip of the teeth contain the highest quantity of trace element when compared to other regions of the mandibles. And here is the same result, but I show in the form of atomic percentage. The previous one is the percent by weight. And next, I also use statistical analysis to compare the differences between each region of the mandible in terms of elemental composition. And it indicated that it is significantly different between region of the mandible in terms of elemental composition. And I also compare the quantity of each element by element, and the results show that sodium, choline, potassium, calcium, and nickel are the elements that are significantly different between each region of the mandible. So from my study, it showed that cuticle of staphylo also contain trace element and also transition metal as well, especially nickel with a high quantity. In the case of nickel, in other insects, there have never been report about the presence of nickel in cuticle of other insects before, but I detected nickel in the cuticle of staphylo. But in my study, I didn't find or detect manganese or zinc in the cuticle of staphylo at all. So from my result, from my study, this leads to the question, what are these elements from? Even though in my study, I didn't conduct the experiment to examine the history or the original source of this element, but I believe that it should be from wood or food that they eat during the larval stage. That because in all insects, not just only staphylo, the cuticle, the material that used for synthesize cuticle usually come from the material or biomolecule that insect accumulated during their larval stage. And in the case of staphylo, during their larval stage, their larvae feed on wood in decaying logs. So I believe that these elements also metal come from the wood that they eat during larval stage. And the next question is, what is the function of trace element and metal found in the cuticle? I would like to say that I do not know yet because there is no any report about this before, but there are two possible explanations. The first one, as I mentioned earlier in the introduction, that some metals can increase the hardness and mechanical strength to the cuticle of insects. And another possible explanation, it is about the detoxification, because some metals may be toxic to insects after eating. So some insect may develop some mechanism to reduce toxicity of this metal by turning it into inactive form and then accumulate in the cuticle. So for my future work, I would like to test for hardness using nano indentation to find the relationship or association between the accumulation of metals and also the mechanical property, and also expand my study with other insect species as well. Finally, I would like to thank the Asahi Glass Foundation and Faculty of Science, Chulalongkorn University, and also Research Equipment Center, Chulalongkorn University. Thank you very much.
Now it's time for questions.
Okay, thank you for your presentation. And I'm interested in the difference of the types and the beetle. And please let me know your opinion. As you know, I mentioned the next future, next plan. You are searching another beetle. Is there any correlation between the type of the beetle and the specific metal element found in the cuticle? This means how does metal the elemental deposition vary between different beetle the specific? Your research is the same the species, but another type.
I don't know what happened with. Okay. Yeah. You mean the differences between each type of beetle?
Yes.
Okay. Unfortunately, because I got with a few number of individual of each type of the stab beetle, so in my analysis, I combine the data, the elemental composition of all types of the beetle together and compared the elemental composition between each region of the mandible instead. But I didn't compare the difference between each type of beetle because I got just only few number of individuals. So it cannot conduct the statistical analysis to compare it.
Okay, I see. Thank you so much.
Thank you.
Thank you very much for interesting research.
Thank you.
If you compare male and female, their larval stage, they are eating same food, I believe. But in case of male, some metal like nickel is concentrated to his weapon.
Yes.
Oh.
I confess I do not know yet how it accumulated in the cuticle of beetle because there is no any report about this before. But if we cite from previous study in other insects, mostly they usually focus on the presence of zinc in the cuticle, and they report that the zinc will attach or have chemical bonding with amino acid residue or protein in the cuticle. But for other metal, we do not know yet.
Thank you.
Any other questions? I'm wondering about the amount of the metal that you find. I'm not sure whether you measure the cuticle in other parts of the body, right? So I'm not sure whether this metal also found in cuticle from other parts of the body, and or it's only like maybe found more in the mandibles.
That will be my future research, but in this study, I didn't do that yet.
Okay, and what about the level of these metals? Like if you collect the insects from like you said different regions, and especially if the level depends on the food that they eat, then do you see a lot of like differences in these metals from insects from different like regions or different locations?
I think it is possible like that, but there is no any proof to prove that hypothesis. But I think it is possible like that. But currently I do not know yet. But it is possible.
Yeah. Thank you. Any other questions?
Okay, I have another question. I think that there is several methods to assess the strengths, such as compressive strength or the tensile strength and so on. Why does to adopt the nano indenter the next future the research?
Can you repeat it again?
Okay, please show me the next research, the final the last page. The future analysis. Okay. Previous, yes, examine the weapon. Sorry, the previous page. Yeah, this one. Yeah, testing the hardness using the nano indentation. But I think that there is several types to examine assess the strength, the compressing. Tensile and so on.
Yes, nano indentation is a technique by compress the tester into the specimen and measure the hardness, and it can provide some physical properties such as young modulus or something related to the mechanical property. So it can tell us about the hardness of the cuticle and also other physical property as well.
Okay, I got it. Thank you so much.
Thank you.
Okay, if there's no more question, thank you so much, Ajahn Nat.
Thank you, Hab.
Okay, let's move to the next speaker. Ajahn Dr. Nathapol Pombupha, also from the Faculty of Science. He's from the Department of Microbiology. His talk will be on the investigation of biocrust by metagenomics and exometabolomics. Please welcome Ajahn Nathapol.
Okay, good afternoon, everyone. Thank you for coming to the talk. So today I would like to take you to another side of the world. I'll be talking about the biocrust. You might be wondering what it is. So let's take a look at this news, which is kind of making on the science recently. So the biocrust has been kind of reported that it's been protecting the Great Wall of China. So it has actually been kind of identified as the living skin. Okay, so what does that mean? Well, taking a look here. So the first question is, why do we even care about, this biocrust at all? So the first thing is, these biocrusts, they have been found in the drylands, but why drylands? Well, if you have not noticed this yet, the Earth land surface is actually covered by around 40% of the drylands. And it has been estimated by, in the next 20, 30 years that this could be increased significantly into 50%. So that means half the Earth land surface could become dryland very soon. And the biocrust itself is actually one of the major components in these drylands. Turns out if you take a look at the floor in these pictures, you might not actually see it, but the soil surface is actually covered by these biocrusts, and it could be up to 70% of these land surface. Okay, so this is why biocrust is actually very, very important in these drylands. Now, so if you take a look at this soil surface, it may just look like the dirt, right? The picture on the left, you see this dry biocrust. Turns out, the researcher have been taking a look at this. It's actually the combinations of different microorganisms, including cyanobacteria, those kind of green guys, eukaryotic algae, fungi, lichens, even biophytes in some cases. And they actually live together and form a community that kind of protect the dryland soil surface, and these kind of forming the crust layers on these kind of top millimeters of the dryland soils. Now, why do we think biocrust is important for the dryland soils? Well, if you take a look at these two pictures, actually coming from the very same place. It's just different areas of that particular regions. On the left side, you see the picture of the biocrust covering the soil surface, those kind of darker colors. Like I said before, it has been recognized as the living skin of the drylands already. This is mainly because those microbes that we see, and they kind of contribute to nutrient cycles in the soils, stabilize the soil surface, capturing the dust, and also absorbing water. Because in drylands, there are only very few rain events. But on the right side, what do we have if we living in the area without the biocrust? Well, actually, you will see this kind of loose sands, the dust problems, and that's creating respiratory disorder in the populations that living near this area. And turns out in the U.S., United States, there has been an increasing of this respiratory disorder about 70% for the last couple of years. And in some cases, you might have to spend $50,000 just to treat this respiratory disorder. And that is why the biocrust is very important for the drylands. Now, you might be thinking that, well, this is, the problem of the U.S. or somewhere else that, might have these drylands. Turns out it might not be the problem for just that, particular place. On the left side, you see this dust storms actually being blown away from, one place, which is kind of quite far from the town, but it can actually cover the entire town. And on the right side, you see the satellite image showing that the dust can be blown away from one continent to another continent. So, even though Thailand or in this area we don't really have this big dry lands or dry land per se, but we might actually get this effect if we have this dry land expanding. Now, for us, why does it matter? Well, as of right now, what we can actually do is we want to save these biocrusts, but what we can do is actually number two, trying to tell people who coming to national park or these dry lands, telling them that you should not bust these crusts because they actually alive. They contain these microbes. But for the Asahi Glass Foundation Research Fund, we actually are able to expand more on the restorations and management, trying to look for what are these microbes, what they can do, and when are they growing and why. So today I'll be showing some of the results that we get from this funding. First of all, what we know so far is that these microbes are actually kind of different from geographic region. Even though we focus on these pictures, it's actually from the Mojave Desert in California and Nevada in United States. Turns out, even though they're in the same desert, it's actually showing you that the microbial makeups or the microbes that living within these biocrusts can actually be quite different, even though the human kind of draw the lines that this is the Mojave Desert, but turns out the microbes maybe not agree to what we establish. And from the grant, we actually look further to trying to identify these overall microbial compositions. Turns out, not just what we see in the last slide, we actually comparing what we call diversity analysis. So these dots showing you that if they're closer together, that means the microbial makeup or composition is actually very similar. Turns out, even though we identify them as the Mojave Desert, in the black dots, they are at the southernmost part of the Mojave. They're actually showing different microbial makeup. Okay? So what does that mean for us? Well, we do know that it may look the same, look just like the soils, but it doesn't mean the microbial makeup will be the same. Okay? So from the Asahi Glass Foundation research, basically we look further into the dynamics because we now know a little bit about who they are. We want to know how they change because we do know that they are probably active when they get the rain events. So basically we already have the data on when they're kind of in dormant stage. Now we want to take a look on the right side when they get the rain or the water activations. How do they contribute to these microbial changes? Well, from this research, we focus on the two terms. The first one we call the microbiomes or the microbial compositions. And the second one is the exometabolomics, which means the metabolites or the materials that these microbes produce. Okay? And we want to take a look at, okay, all right. We want to take a look at the dynamics. But however, in these area, there are actually 5 different kind of biocrusts, but there are only the 2 major one, which is the light algal crust and the cyanolichen crust. So the one that look just like bare soils and the one that have kind of darker color of the lichen on it. So basically, we want to take a look at how they change after we get these water activations. So we take a look at 3 minutes until 96 hours after water activations. And turns out when we take a look here, the very key thing that we find, you see that kind of eclipse, the left and the right, it's showing you that the crust types is actually very, very different. So the lichen ones actually have very different microbial makeup comparing to the kind of the one that look like a bare soils. And the other one is we take a look not only at one type of the microbes, we take a look for bacteria and the fungi. It turns out the pattern might not be the same, but we actually see this different between the crust types. And we also see that the metabolites or the substance that they produce, they kind of having this 4 different cluster. So you see that after they get water activations, they kind of respond. We don't know the first one yet. We're trying to find out. But basically, they respond to the stress. Those waters seem to be stressing these microbes, and then they kind of grow, and then they develop this kind of specialized pathway. Okay, so what does that mean from this research? Well, first of all, we kind of do know that even though there seems to be the water seems to be the source of their activations, but it seems to also be the source of the stress for these microbes as well. And they also kind of contribute to the changes in these microbial communities. And we now have confirmed that the biocrust type that we see in these regions is actually quite different in terms of the microbial makeup. And then lastly, we do see that the metabolites that they produce actually changes over time, okay? So that means in terms of restorations and management, these might be the key that we have to take a look at, what are the microbes are actually producing and how we can induce them to produce this and grow as the communities. Okay, and lastly, I would like to thank the Asahi Glass Foundation along with other grant funding that we received. This is part of the bigger grant, and right now we're trying to finish up this project in collaboration with Dr. Suzuki from University of Tokyo. Take a look at the metabolites, the last part. And I would like to thank my lab as well in kind of working on this project and finally reaching the last part of the project already. Thank you.
Thank you. Thank you, Ajahn Nathapol. Question from the floor.
Okay, thank you for your presentation. I'm not familiar with this area, so then I have the simple question. So, are there the specific the metabolite that are produced only after water activation?
Well, as of right now, even though I like to answer your question very directly, but we still working on that part to finalize with Dr. Suzuki. So I can't give you the definite answer yet, but as far as we know, there are 4 patterns of those. By the first one, we don't know what that means yet. There kind of a lot of variations. We don't know if it's just the noise or not. But the pattern that we see is the second, the third, and the fourth that we see stress response, and then the growth, and then spatial light pathway. But in terms of specific metabolites, right now we're trying to find out about that yet, but we don't have the final answer for you yet. Sorry about that.
Okay, thank you so much. I appreciate it.
Okay. So I'm not sure, but if I compare with stable biocluster and the, how should I say, which has adaptability for the environment change biocluster, 2 kind of biocluster. So if we think about the rapid climate change, I think the biocluster which has adaptability only survives on the Earth. Is it true?
Well, I'm not totally sure if I can answer that as well. But basically, based on the research that we know, one of the things why we study them is because they grow very slowly. In terms of adaptations, it's actually quite hard, even though they live in dry land itself, and it's very harsh environment. Turns out, like for some of these, there have been monitoring that some of them actually grow like in size of like centimeters per year. So it's actually quite slow, very, very slow. So that means in reality, if the dry land is expanding, it might be the case where they cannot adapt or cannot expand at the same rate as the dry land itself. So we might end up having those kind of loose end problems or kind of dust storm. So, well, I think right now we're trying to figure out, I guess, to help them adapt, I guess, but we don't have the final solution yet because my collaborators has been trying this in the field, but they grow, when we add water, but then, it's not natural. So that means after they kind of losing water, they die.
Very important. Thank you.
Yeah.
I have one question because I'm not in the fields of the natural science, so I would like to know that how can you differentiate between just the soil and the the crust? Yeah, how can you differentiate it?
Okay, so basically, like I said earlier, so these are the top millimeters. So that means when you pick, typically in Thailand, right, or in our area, if you pick up the soil, this will be just like bulk soil, right? But in those areas, if you pick up the soil, you will see that the top layer is actually separated from the soil underneath. So you can see that top kind of millimeters being kind of one pack of the crust. So it's very, very different from the soil itself.
Maybe we have some time, right? I'm wondering, because I'm working on plant, right, and like stress, salt stress, drought. I'm wondering whether would it also be interesting to look at the metabolite profile when it's getting dry, because now you like it had more water, right? Activate with water, but sometimes it's gonna get dry, right?
Right, exactly. That's very good questions, and we actually like to try that, but based on the funding that we have, so it's very limited. So right now we focus on what they secreted. So that's why we say it's exometabolites. But definitely, right now we take a look at the first phase that they get activation, which is the water, right? But I think the next phase that we have to take a look at is how they go back to their dormant stage. I think that's also very, very critical. But right now, I don't have the answer to that question as well.
Okay, so thank you so much, Ajahn Natapon.
Thank you very much.
Okay, now we move back to the medical field. The next speaker is Assistant Professor Dr. Patharin Tangthana Takul. She's from the Department of Transfusion Medicine and Clinical Microbiology, and her talk today will be on STAT4 regulatory pathway in type 1 interferon inducible monocyte-derived dendritic cells.
Yeah. Good evening, everyone, and good evening to the extreme member of the Asahi Glass Foundation. And I would like to extend my deep gratitude to the foundation for the generous support on my research project. And today I'm very excited to present the progress and the key finding of my study that study in the STAT4 regulation pathway in the type 1 interferon human dendritic cells and autoimmune disease models. And let me introduce myself a little bit, but I'm the immunologist, so I will focus in the immune cells and will be the very deep science today for the today presentation, and I'll take you to my journey of my research that I've got a lot of obstacle and many solving like that. So in the beginning, I would like to provide you a brief introductions about my research work that mostly I'm interested in the STAT4 transcription factors, which is the important in the immune cells that is critical in the immune regulation. The disease model that I'm interested is the SLE disease, which is the chronic inflammatory autoimmune disease that our immune cells will mistaken attack ourselves. And the STAT4 transcription factor is actually has been found as associated with the disease. So it means that this transcription factor may drive the disease or drive the disease pathogenesis. And basically, we know that this transcription factor is the transcription factor downstream of the interleukin 12. The interleukin 12 is actually enhanced the T helper T cells in our immune system that will help to kill the virus or kill the pathogen. So, and very interesting, they found that the function of STAT4 is in each immune cell is different. And is related to the immune disease. So in the T cell, we found that STAT4 is increased the anti-apoptotic genes. It means that STAT4 increased T cell proliferation. T cell is very important in the immune system. Another one is the STAT4 increase the interleukin 21 that can increase the autoreactive antibody. So autoreactive antibody also drives our the immune system into the disease. And next one is the NK cell. They found that STAT4 increase the perforance enzyme that can kill the cell or kill the microbe. Yeah. Then the last one is the macrophages. They found that STAT4 can increase the IL-12 and induce the interferon gamma, which is the main cytokine that increase the killing activity and inflammatory activity. For the last one is the monocyte-derived dendritic cells in mouse, and they found that STAT4 can be inducible by the interferon alpha with unknown function. So recently published in the very high impact journal just this year, they found that STAT4, when they knock out STAT4 from the mouse that conditionally knock out STAT4 in the dendritic cell and in T cell, they found that mouse will not get the disease, will not get the experimental autoimmune encephalitis disease. So it's very important that STAT4 can really associate with the pathogenesis. And when they adoptive transfer back STAT4 into the mouse, they found that mouse get the disease. So it means that STAT4 in dendritic cell is very important and associated with the disease development. And they found that they hypothesized that STAT4 may regulate the dendritic cells, mouse dendritic cell migration into the inflammatory. But the STAT4 study in human is underexplored. So that leads to my research question about the STAT4 in human dendritic cells. But before we jump into the research, then will introduce you to the human peripheral blood dendritic cells that is very low percentage in our blood circulation, but very, very important. The human dendritic cell will normally differentiate into two subclasses. That is the plasmacytoid dendritic cells and conventional dendritic cells. And dendritic cell is the only innate immune cell that can communicate with the adaptive immune cell, that is T cells. Conventional dendritic cells, normally I will call DC, dendritic cell DC. So it will divide into three main classes now. And the additional one is the dendritic type 3 that just recently published in the Nature, that is very important in the immune system. And previously they found that the dendritic cells already predominate in the SLE patient, and it can be the interferon type 1. This one is the interferon type 1, interferon beta, that found in the autoimmune disease and induce the maturations of the dendritic cells, especially conventional dendritic cell type 2 and 3. And it's also induced another type of T helper cell called T helper 17 that important in the inflammation. So in my research concept, we would like to study the it's not going. We would like to study the the inducible STAT4 in human dendritic cell that no one haven't studied yet. So we have to research question whether STAT4 can be inducible in human dendritic cells, and what is the role of the STAT4 in human dendritic cell. So there's two major research question in our research. So first of all, I have to pass to the ethical process that is take me very long time. And finally, I get the ethical approved to get the Buffy code from a healthy donor, 11 people, and then I purify the peripheral blood and get the PBMC. Then get the dendritic cells, and then we study the STAT4 expression through the real-time PCR and Western blotting. Then in the same time, I'm also study the response of the cells to the interferon beta. And in order to characterize the role of STAT4, I'm trying to inhibit the STAT4 expression using the chemical reagents called lysophilide or LSF here. And then try to study the role of STAT4. And to confirm that, I'm also study the role of STAT4 using the chromatin immunoprecipitation sequencing. And first, I'm sorting the dendritic cells and using flow cytometry, and we actually get the conventional and plasmacytoid dendritic cells, and we subclass the conventional dendritic cells into dendritic cell 2 and 3 and dendritic cell 1, and percentage of the dendritic cell is roughly the same as the previous publication. So actually we successfully separate the dendritic cells from the healthy donor. Then we investigate the STAT4 activation. Actually, first we activate the cell with IL-12, which is the known stimulator for STAT4. But unexpected, we found that IL-12 could not induce the STAT4 phosphorylation. That very unexpected. And we found that interferon beta actually induced the STAT4 phosphorylation very clear. So unlike in mice, in human we found that IL-12 doesn't play the important role to activate the STAT4, but interferon beta is more potent to activate STAT4 activation. And then we use the interferon stimulation, and we check whether the interferon can induce the DC cell maturation. Then we found that this is actually matured by, we measure the maturation factor, this too, by flow cytometry. And then we also measure the cytokine release from the culture supernatant using the very high technology screening using the Luminex technique. And we found that actually interleukin 17 and interleukin 23 and many inflammatory cytokine already changed in the specifically subpopulation of dendritic cell, which is the conventional dendritic cell type 2 and 3. And then we go further, we study the STAT4 expression and potential downstream target of STAT4. We actually found that STAT4 can be inducible in the human dendritic cells, especially in the human dendritic cell conventional type 2 and 3. And another potential downstream genes also increase. But when you see the IL-12, it's actually down-regulate in human dendritic cells. So it means that maybe IL-12 is not in human is not actually downstream of the STAT4. And we confirm also from the gene expression, we also confirm by using the Western blotting. And we found that the inducible STAT4 actually increase in the human dendritic cells. And this is the first is a novel finding in our study. So we also measure to verify the expressions of potential genes of STAT4 in human dendritic cells. It's not passed. Using the kinetic dependent manner. Go back, please. Yeah, and we also found that the interleukin 23s and interferon gamma were significantly increased after we induced the human dendritic cells with the interferon beta. So actually IL-23 is very important to enhance the T helper 17, which is important in the inflammatory response. Yeah. Then we characterize the role of the STAT4 in human dendritic cells using the, previously I say is the lysophili to inhibit. It's previously known like an anti-inflammatory agent to treat the type 1 diabetes and specifically block the IL-12 induced STAT4 signaling from previous literature review. And we expect that we will got the STAT4 phosphorylation inhibition like that. So we study the phosphorylation inhibition using the lysophili. Then we measure the cell viability after treating this chemical, and this chemical doesn't affect the cell viability. But with the IL-12, we doesn't see the inhibition of the lysophili after we treat it with the varied concentration and also with the interferon results. And also when we vary it with the time of the blocking with the lysophili, we also didn't get the expected results. So actually we have tried a lot to see whether lysophili can really block our STAT4, but actually lysophili cannot block our STAT4. So we moved into another experimental to try to characterize the role of STAT4 in human dendritic cells. Using the chromatin immunoprecipitation, the STAT4 will bind to the DNA, and we use the antibody to precipitate the STAT4 and use the DNA to sequence using high throughput sequencing. Then we do the analysis to see whether what is the genes that STAT4 really bind. And we found that after we stimulation, we found the quite difference of the result between stimulation with interferon and unstimulation. And we actually study the model of binding, which is look normal. And we annotate the genes that STAT4 really bind, and is mostly bind on the promoter regions of the genes, and mostly in the transcription start site. So it looks like that our data is normal. And when we do the pathway analysis of the binding sites, we quite surprised that we got the pathway that STAT4 regulate the protein couple of the carbohydrate transporter and the transport of the carbohydrate protein to the Gonzai. So to be very surprised to me that STAT4 can regulate the cell metabolism. Yeah, so our key finding is that we identify the inducible STAT4 in the human dendritic cells that specific population in conventional DC type 2 and 3 by interferon. And unlike in mouse, the interferon beta is more potent to enhance human dendritic cell maturation than IL-12. And in human disease, it seems like lysophilide may not directly inhibit the phosphorylation of STAT4. And in the right now we still try to study the regulation of STAT4 in human dendritic cells. That further experiment may be needed. We may need the PCR reaction to see the genes after STAT4 binding. And we partially published this in the Journal of Immunology. And thanks to the funding support from the Asahi Glass Foundation to let me start this project, because before get the funding, I haven't started anything yet. And thanks to the member in my team, the Excellence Center of Immunology and Immune-Mediated Disease, and thank you to the master degree student that helped me with this project. Thank you very much, Kai.
Thank you. Okay, so now the floor is open. Any questions or comments? Please.
Yeah, very long story.
Very primitive question.
Sorry?
Yeah, very primitive question, sorry. But you mentioned that the difference between mice and human
Yeah.
case. So what is the reason why the difference?
What is the reason of this difference? I think the signaling pathway of the interleukin 12 to induce the STAT4 in mouse and in human may be different, because basically dendritic cell is very hard to study. It's very low percent, as you can see, it's the 0.5 to 2% in the human blood circulation. So if you draw the blood from normal human like normal like us, then we got very low number of the dendritic cell. So mostly people study the dendritic cell from macrophages to induce it and then change its phenotype into dendritic cells. So, but in my study, we directly purify the human dendritic cells from the bulk, from the healthy blood donor. That we got a lot of the Buffy code, a lot, 800 million cells from the donor. Then we get enough dendritic cell to study. So I think this is the difference between the mouse and the human dendritic cell that may have the different signaling pathway. And actually, this signaling pathway, they try to characterize in order to decide the new therapeutic target to directly target to the dendritic cells and then prevent the disease occur.
Okay.
Thank you very much.
Any other questions?
Yeah.
Okay then. Thank you for really informative talk.
Yeah, thank you very much.
Thank you so much. Okay, now our so we have our next speaker, Assistant Professor Dr. Supansa Yodmyeong, whose talk will be on the development of imitation shark fins by tissue engineering technology. She is from the Research Affairs Faculty of Medicine. Let's welcome Dr. Supansa.
Thank you for opportunity to come to this room and speaking and talking about our research project. I would like to introduce myself a little bit. I have a background in biomedical engineering, but I work in the Faculty of Medicine. So basically, my research background is all about making the tissues, cartilage, bone, and everything. So I think the Asahi Glass Foundation helped us to expand more, like challenging plus fun projects. So we make a cartilage from, like base knowledge to be some food project here. Thank you very much. In the first, yeah. So what is that? Can you help me with? in the first line. All right. I can point it here. Okay, so shark fins are very popular delicacy food in Asia because with the belief that the shark is a majestic animal, so it's strong and it's tough, have a very difficult life in the ocean. So maybe when we consume part of their body, will make us strong as well. So shark fin will symbolize the whale and the hell as well. So you can see through the shark fin soup. But however, the harvesting process is very, very brutally devastating because the shark will collect into the ship, cut out the fin, and throw the body and the remains back to the oceans. So we kind of like don't want this happen to our society. So that's why we develop this kind of project. Maybe it's alternative options for making the shark fin soup. So in terms of the health concern, you can see that shark lifespan is probably 10 or 30 years up into the ocean. So they have a higher chance to expose all the pollution, heavy metals and other things. So it's accumulating into the meat. Like other apex animal, tuna, seals and other things, but we not consume seals. So all the big fish in the ocean will have a higher chance to accumulate the mercury into the meat. And also when we take a look at the nutrition facts of 100 grams of shark fin soup, basically it's very low calorie, 46 calorie, and have only very small trace amount of protein, 3 grams. And on the other way, so you will see the imitation shark fin in the market. So there's no protein at all because most of the key ingredients from seaweed. So basically it's polysaccharide. But some seaweed, probably our body can digest it. So basically the calorie we get is around 300. So there's no protein, but make you fat, something like that. So it's go to the right direction already, not killing the shark, but it doesn't have enough nutrition value. So we try to close this gap by maybe we can use the already existing pork industry, because the pork will be everywhere in the market, in the food that we see everywhere, like a hot dog, salami, sausages, and also the Thai meatball. So it's already there. And the next question is that why we not converted pork meat into the shark fins. So what we are trying to do is that we're trying to transform the pig cells into the shark tissues. So basically, it's the fin. So we isolated the cells from the pig ears. Is not very easy step, but we know how to do it. It's a little bit challenging, but we can do it. We isolate the pig cells, which called chondrocyte. This is the precursor for making cartilage. And also we mix them with the hydrogel. So in this context, we call bio-ink. And it's edible. So we not use the random hydrogel we cannot eat. We mix them together and we load into the syringe to print them by using the 3D bioprinter. As you can see, I will elaborate about this part later. And the shape that we want to print is the brush-like structure, like the fin and the like a hair structures. So. Not here. Yeah, so in focus on to what's going on in the petri dish. We mix the bio-ink with the cells. And then for the ink itself, it's all edible. For example, alginate, gelatin, and xanthan gums. So it's already in the food additive. For example, xanthan gum we see everywhere in ice cream, in cookie, because it's thickening agent. And also for the alginate, we probably familiar with the bubble tea. Yeah, all that thing is in there, and also gelatin and jelly. So we combine all these ingredients into different ratio, and print it out into the bath. And the bath is made from gel as well, because we print it out by hand. Like this, it cannot control the shapes and it's flat and wobbling. So we use the CAD software to draw something look like the fin shape as a prototype, to test the concept. And we print into the gel. So we add some color, the red color, for the visualizations. And we have our custom-made bioprinter named Enterprise 3.0. We have 1.0 already, so we move a little bit forward, change this and that. So we got our the third version. So this is something for some a hint of what we are doing. In terms of scientific approach, we focus on the edible hydrogel or the ink because we have a lot of ingredients combined there. We vary the percentage of xanthan gums, and we found that because it's the key element, so if it's too much, it will dissolve very fast, like the line 1.5%. Starting from 300, it swells so much and degrades right away in first week, second week. But when we minimize the percentage of xanthan gum to 1.5 or 2, so definitely it swells from 100. And then at the point at 3 weeks, it stops there and degrades. So it provides us the sufficient time to incorporate all the cells, and the cell have the time to secrete the collagens or extracellular matrix. So we will keep in the 2% weight by volume of xanthan gum into our ink. And we also measure the viscosity of the gels. So to make it short, you will see that 2.5%, there's a line of the green and the blue crosses together, and it's very far from the right, which means it's more solid-like elements compared to the 1.5, so it's more on the left side, so it's more liquid. So we choose 2.5 in our experiment to make the fin, the cartilage. And this is the quick view of 1 month or 28 days. In the cross section at the end, you will see the shape is round and there are cells in there, maybe a different focal plane. We cannot see everything at once. And on the side, you will see there's a green live cell, represent live cells on the side. And also this is some fluorescent microscope and bright field microscope. So the cell is there. And actually, we want to make it better. So the gel is very fragile, actually. Because think about that, it's fair enough because chalk will take 10 or 20 years to develop the fin, but we have only 1 month to make it. So we will struggle with how to improve the chewiness or more rubbery. So we will work on it after the project is over. But actually, the science behind this technology is tissue engineering. So we combine all the things together, but it has 3 elements, which is we call triad. The triad, 3 things. Cells will live within the biomaterials. In this case, it's the bio-ink. And then we use the external environment, which is architecture, how to fabricate the shapes, and also how to incorporate the stimuli, the nutrients in our culture system. Okay, but the take-home message, then I want to raise some awareness and something maybe is look crazy. How about we transform, translate what's going on into the lab to the kitchen? So by using the food grade rather than the research grade, for example, the cell definitely we can isolate it from the lab, but when put or mix them together with food ingredients, xanthan gum, gelatins, alginates, and then we can make it maybe at home at a sterile condition rather than we use the research grade, which is red elements like phenol red, have some fetal bovine serum, some buffers, some antibiotics. And I don't know, is it possible if we have a dashi soup because it has amino acid, it's made from seaweeds. already. So maybe we can grow some kind of shark fin in the kitchen, in the food level. Yeah. And also, last but not least, so our research presents a sustainable alternative to the traditional shark fin harvesting by using the tissue engineering and 3D printing. And this is our lab member. Yeah, so our main job is making chondrocyte and cartilage disc, and also the chondrocyte and cartilage in the trachea as well, believe it or not. So because we have the cartilage ring around the windpipe. And this is my lab member, postdoc, master, and PhD students. And also our project is related to making the biomaterials, tissue engineering, and try to make a green tissue culture, minimize the chemical in the lab. And thank you very much, especially the Asahi Glass Foundation. This is all my talk, and I'm ready to take questions and comments. Thank you.
Thank you so much, Ajahn Supansa. Very futuristic. Okay, any questions, comments from the floor?
Okay, thank you for your presentation. I'm interested in the structure, the imitation structure, the property. And how do the structural property of the imitation fin comparing with the real shark fin?
Yeah. So actually, we bought the shark fin from Chinatown to see what it looks like. It's look like a broom, like all the fins. So it's not very elastic, but it's more fragile. But not fragile. So it's tough like like a broom, actually, because we can bend, but it doesn't break. But our gel still break and a little bit brittle, maybe because of we have only 1 month to grow, but the shark is take entire their life. Yeah, so it's have some more work to do on that part.
Thank you so much.
Yeah.
Thank you so much. I think the gel-in, gel 3D printing looks very important for your process. So let me know why the advantage of the gel-in, gel process.
Right. Yeah. Okay. Very good questions. We try to print it out by using hand or using the flat surface. What we found that it's melt right away. The gel is, when it's very soft, the ink is very soft, it's flat right away on the petri dish. But when we have the support system, which is not completely liquid, but it's gel like a dishwashing soap like that, like a honey, yeah. So it support our ink stay in the cylindrical shape along the way. So it's not flat or spread out, yeah.
Patients. No. So in your abstract, you mentioned about like the experiment that maybe took like 4 weeks of culture. I'm wondering at that point how much cells do you have? Like, because at the end you want you probably don't want the alginate or the gelatin, a lot of it, right? At the end, you want the cell, the tissue, right?
Right. So the target, the ideal tissue that we want at the end to produce, the cell will produce a lot of collagen. But most of the tissue that we get mostly are glycosaminoglycans or GAC. Yeah, so it still absorb the water. It can expand. But when we have the collagen, it's more like fibrous structure, so it's hold the shape of the gel together rather than swollen. So yeah, the ideal is the collagen is still suboptimal, but GAC, yeah, we made it. Glycosaminoglycan, we make it.
So when successful, you think like how long it would take to have that kind of structure?
Well, I will try 2 months first, and in the meantime, we probably have to optimize the what is called the amino acids ratio or the formula of in the culture media. So maybe they need more proline or some different amino acid which contribute to make the collagen rather than regular amino acids. So there's a few optimizations which take times, and also the length of culture period, the formulation and the times.
Right.
So I would say 2 months, but this is just 1 month.
Okay. Very interesting. Thank you so much for the talk. Okay. Now we have Associate Professor Dr. Supawadi Umthong. She's from the Department of Biochemistry and Microbiology, Faculty of Pharmaceutical Sciences. Her talk will be on the characterization of human and bat march to protein in restriction of viral envelope glycoproteins. So please welcome Dr. Supawadi.
Okay, good afternoon, everyone. So thank you so much for this opportunity for me to present my research under the Asahi Glass Foundation. So I started my career at Long University from January 2022, so not long ago. So at that time, I finished my PhD and postdoctoral training in the US, and then I moved back for good to Thailand. So my research during my PhD and postdoc, we focused on host pathogen interactions. So as you know that pathogens are organisms that can cause diseases in otherwise healthy hosts. This include the bacteria, parasites, fungi, viruses, and prions. And upon infection, our immune system is critical to detect and eradicate the pathogens. So, and the pathogen that we study in the lab is the retrovirus. And retrovirus is the envelope single-stranded RNA positive sense virus. And and you can see the structure of retroviruses that they have the upper part, which is the surface envelope or the SU protein. And the bottom part is the transmembrane envelope protein or the TM. And apart from that, the virus also have the structural such as membrane and also some structural proteins such as the matrix protein, capsid protein, and they have the RNA as the genome that bound to nucleocapsid protein. And other than that, virus also have integrase enzyme, reverse transcriptase, and protease enzyme. Yeah, just go ahead. Go ahead. Yeah, go forward. Just move forward. Yes. So for the life cycle of the virus, it starts by the binding of the host cell receptor, and then the fusion will be happen. Okay, and then the virus will start the uncoating process, and then the reverse transcription to change the RNA into the cDNA. And after that, the virus will the genome of the virus will move into the nucleus and incorporate the genome into the host genome. And after that, the virus will start the transcription and translation process, and then package as the virion to infect other susceptible host cells. Yes. So we focus on the immunity that counteract during the viral infection, and the immunity that I particularly interested in is the intrinsic immunity here. Okay, so the intrinsic immunity is actually for viral infection is the first frontline defense against viral infection. As you can see that when the virus infection happen, the intrinsic immunity can screen the virus and reduce the infectivity of the virus after it's passing on to the innate and adaptive immunity. And the type of intrinsic immunity that I particularly interested in is the cell host restriction factor here. As you can see that here showing you the host antiviral protein or host intrinsic immunity that counteract with the viral protein. And this has been evolved together for a very long period of time during the evolutionary scale. So the host antiviral protein is it's act as a frontline defense against viral infection, and it stops viral replication and transmission, and is usually upregulated by the interferon type 1, which is immunity against viral infection. So in this here, showing you the life cycle of HIV-1 that I explained to you before, but you can see that there are so many host restriction factors inside the cell, such as TRIM5 alpha, SAM-HD1, MX2, upper-back 3G, tetherin here that can target HIV-1 at each step during their life cycle. For example, TRIM5 alpha can stop the uncoating process. SAM-HD1 and upper-back 3G can stop the reverse transcription process. And viral also have their viral proteins such as VPU and VIP that can counteract with the host antiviral activity. So this is like a tug of war between the host protein and viral protein. And here showing you the HIV, when they produce the HIV particle, so the envelope of HIV will synthesize and translocate to the cell membrane. And currently there is a group of protein called MASH protein that have been identified to target HIV envelope. So MASH protein is the membrane-associated ring CH type finger proteins. They are E3 ubiquitin ligases enzyme, and they contain the ring CH domain. As you can see here, this is the ring CH domain in the MASH protein. And because it's the E3 ligase, so it can bind to E2 ubiquitinated. And then, so the MASH protein, actually in our body has 11 types of MASH protein, but those that are antiviral have been identified to be MASH1, MASH2, and MASH8. Okay, so inside the cell in general, MASH is located on the cell membrane. It has the ring CH domain, and it can transfer the ubiquitin from the E2 to the substrate. And this leads to the degradation of the cellular receptor that can be targeted by MASH protein. So this protein can get degraded by MASH protein. Okay, and we know that HIV envelope also located on the cell membrane, and we actually have been identified that MASH protein can actually target HIV envelope and lead to the degradation of the envelope of HIV-1. As you can see in this picture here, when the HIV envelope is synthesized and translocate to the cell membrane, and the virus will get packaged and released as an infectious particle. But with MASH protein present on the cell membrane, it can reduce the envelope on the cell surface, and this resulting in the reduced envelope viral particle, and the overall infection is reduced. So we published this a couple years back in MBio, telling about the MASH activity that target HIV-1. And apart from the HIV-1 that is belong to retroviruses, we also particularly interested in those that have enveloped glycoproteins, such as this group of virus, and also this group of virus that have the envelope. So we asked if they actually target just only retrovirus or other virus as well. So we did some experiment, co-transfection of viral envelope glycoprotein with other human MASH, and we perform Western blot to see the protein levels. So you can see here not just only retroviruses, but Ebola glycoprotein, influenza heme agglutinin, and also SARS-CoV-2 spike can be targeted by MASH protein. You can see the band here is lower compared to the empty vector control here. But not all viruses that can be targeted by MASH protein. For example, Zika envelope here cannot be targeted by MASH protein. So we were excited about that result, And one of the question I would like to ask is, like, you all know that the bats harboring so many viruses that can, like, be very serious for human, but those bats doesn't have any symptoms at all. So we ask, why don't bats get sick at all, even though those viruses are very lethal in human? So I want to explore this under the Mass2 project. So, and you can see that so many viruses that are harboring in bats, such as Nipah virus, coronavirus, Hendra virus, and Ebola and rabies. Those are very lethal in human. So this leads me to this project that I want to compare the antiviral activity of human versus bat mask proteins. So simple things that I did was that I aligned bat Mask2 protein and compare them with other mammal Mask2 proteins. You can see the unique amino acid sequence that found in bat Mask2, but this amino acid doesn't show in any other mammal species. So it is very unique in bat Mask2, and it also changes some structure of bat Mask2 compared to other species as well. So what I did was that we have the human Mask2 wild type construct, and I also engineer human Mask2 in the way that it had the 8 amino acid found in bat Mask2 inserted in the human Mask2, and I co-transfect with all the HIV-1 infectious clone, and then I collect the virus and infect into the cells to see if the presence of 8 amino acid will have more antiviral activity or not. Okay, here it's showing you the HIV-1 data. You can see that when we insert 8 amino acid in bat Mask2, it's reduced the envelope of HIV-1 a little bit compared to that human Mask2 that is the wild type one here. And when I compare the infectivity overall, adding this 8 amino acid in human Mask2 reduce the infectivity overall of HIV-1. So it's like very exciting data that we have so far, and we want to explore more what is this 8 amino acid does in bat. So in conclusion, I'm showing you some exciting data here. We are currently studying a lot about Ebola, Nipah, Hendra, and all those that are harboring in bats, and we got some exciting data so far, but I don't have time to show you today. But the conclusion from this project is that we want to explore what does this 8 amino acid do in the and if we can apply this concept into developing the vaccine or some treatment that target or increase the activity of antiviral drug by using the concept of this 8 amino acid. So we just trying to learn from bats to help protect human health. Thank you so much. And this. Yeah, that's my lab, and I would like to thank Asahi Glass Foundation because I did not have much of the grant at the beginning because I just started, and I also would like to thank my department atฬาลงกรณ University.
Okay. Any questions for Ajahn Supawati?
So the Batman doesn't get sick.
I'm bad girl now.
Okay. My question is using this mechanism, how do we improve or application of your result to the actual human?
Mm-hmm. So because the Asahi Glass Foundation, I come up with this idea and we got some data, and I actually applied for further scholarship, and I got the Cornell Juฬาcollaborative funding. So I'm now collaborating with Professor Aguilar Carlino at Cornell University. I actually spent past 3 months joining his lab during summer, and he is very well known about Hendra, Nipah, and Ebola, coronavirus, those that are harboring in bats. So we study all those viruses, And we are trying to identify the binding partners of the host protein that interact with these 8 amino acids so that we know the actual mechanism, like why these 8 amino acids can help the bat to protect themselves from developing diseases.
Okay, one from the back.
Yes, thank you very much. Could you tell me again how MASH recognized its target? And are there any consensus sequence?
So because MASH is the E3 ubiquitin ligases, so they can bind to the E2 that have the ubiquitin. It all depends on whether it can be targeted by MASH or not. Any cellular protein on the membrane, if it can be targeted by MASH, MASH can ubiquitinate that protein and get the protein to degrade.
So by overexpress MASH, you probably destroy a lot of receptor or cellular cell surface proteins.
Okay.
How could it be more specific? Are there any specificity or?
Yeah, because a lot of experiment is done in vitro, and the way we perform experiment, the simple way is to overexpress MASH protein and see if it can reduce the envelope of the viral proteins or not. But we also perform it in the other way to confirm that if just only the endogenous level of MASH in the cell is actually sufficient to block the infection or not. The way we performed that experiment was that we knocked down MASH in the cells by using the lentivirus. So the cell will have very low level of MASH, and then we infect those cells with the virus and see if the infection is increased or not. And we did see that trend in for the case of HIV-1. We perform in T cell line, macrophage cell line, and we also perform in human PBMC. We isolate the human CD4 and also human macrophage, and then we knock down MASH in those cells and infect with HIV-1. And we see that when we knock down MASH too, it increased the infectivity of HIV-1. Am I answering your question?
Thank you.
Any other questions on the floor? Actually, I have one. Actually, regarding the specificity, because correct me if I'm wrong. E3 ligase probably don't recognize the target proteins or receptors by themselves, right? So they probably require other proteins. So in your hypothesis on the mechanism, how it works regarding the specificity to those receptors?
Yeah, so E3 has the domain that target to the targeted protein, right? And also has the domain that bind to the E2. So these two have to corresponding so that it can transfer the ubiquitin to the targeted protein and get the protein to degrade. So we also perform the experiment to, you are asking about mechanism, right? Okay, so MASH protein has the two transmembrane domain on the left and on the right side. And the HIV-1 also have the transmembrane portion. The SU is on the top. The TM is on the past the membrane. So we engineer MASH in the way that they don't have the TM1 or either TM2, and also mapping so many domains of MASH to see which domain is actually recognized and bind to the envelope of HIV-1. And we found that on the TM2, the transmembrane, the second transmembrane of MASH2 is actually interact with the transmembrane of HIV-1 envelope. And this interaction come together when the virus get packaged as a free virion, and MASH go into MASH2 go with the envelope in the virion. Just so yeah, that's one mechanism. So they bind together and then degrade the MASH protein.
Okay, thank you so much.
Thank you.
Okay, if no more question, thank you again, Ajahn Supawadi. Okay, now we come to the last speaker, Dr. Lalita Suriya Arunrod. She's from the Research Affairs, Faculty of Medicine. She will give her presentation on the delay of gratification in non-human primates. Please join me in welcoming Dr. Lalita.
Good afternoon, everyone. I'm more excited than I expected. So today I'm going to talk about the project that we got a really amazing opportunity from the Asahi Glass Foundation. So as I previously worked at a primate center, and I recently moved to the Faculty of Medicine, but I'm still like working on the similar topics, so which is like behavioral and cognitive neuroscience. So for this topic, for this project, it's the starting point of like, it kind of gave us the, sorry, the starting point of series of experiments that we're gonna run in the future. We focus on the self-control, which is a really, is something we face every day, but sometimes we don't know of. So for this project, we, sorry, okay. Excuse me. I can't go backward. Okay. So for this project, it's we have collaboration with like the many primates network, which is like a global network of primatologists, and also we got supported from the Nakhon Ratchasima Zoo, where we conducted our experiments, and also we got some students, interns from Khon Kaen University, which is close to the zoo as well. So I'm gonna tell you how amazing this experiment was. So first of all, we're gonna start with the self-control. So we may think of self-control as like something that can lead to societal problems like addiction or risky sex behavior that leads to HIV addiction, infection, for example. But it's actually what we are facing every day, like binge eating, binge Netflix, and we want to go to the gym, but we skip it all the time. We choose the cake instead of going to the gym, for example. So it's not, sometimes we think that if like we are educated people, we can control ourselves, but many times we are wrong. And the problem is that there's this thing is something we have to blame, which is temporal discounting function. It's a function that devalues the reward over time. It affects like why we choose to spend the money now but not save to the future, why we choose to stay lazy today instead of going and fight for the world or protect the environment, for example. So like, but we can think about this at the individual level, like sugar addiction or alcohol or phone addiction, or even like something that we have to control, like emotions. Sometimes we are so angry and we need to keep calm, and or even sometimes we know that we shouldn't say something, but we did anyway. So these are all self-control problems. Another complexity of self-control is that it's not only about time. Sometimes people think that self-control is like we have to be patient, and in Thai we say angun priao, or we say like อดเปรี้ยวไปกินหวาน yes. In English like better 2 bananas tomorrow than 1 banana today. So if we wait longer for better reward, it's like self-control. But it's more, but the more we think about it, there are many more factors related to that, like effort. Sometimes we prefer having, sometimes we, excuse me. Sometimes we are willing to spend more effort to get more reward as well. Sometimes we work hard, we stay overnight to finish something. Some people have that, but some people don't. And sometimes it's about resist temptation versus maintaining the long-term goal. At the beginning, we decide to go to the gym. We pay a lot of money, but it's so hard to maintain that goal. So what's that, right? It's the maintenance of the goal. And it's sometimes about inhibition or initiatory, which is a really new word for me. Like sometimes we have to resist something to wait for a better reward, but sometimes we have to go out and make more effort to have something better. So this is a really complex equation to think about. If anyone here knows about the marshmallow test. So it's a test, it's a really simple test. Like a teacher come into the room and give a marshmallow to the kid and tell him that if you eat this marshmallow, you can have only one. But if you wait for me to come back in 5 minutes, you can have 2. Some kids managed to do that and some didn't. And Walter Mischel, he did this experiment in 1981, and he followed up for another 18 years and found that the kids who managed to wait have better like academic achievements. But actually, as I said in the previous slide, that it's more complex than that. It's not only the patience, but it's more effort and how to maintain that goal as well, and how you manage the waiting. It's not only waiting, but what you are doing during waiting. And how do we do this in animals then? And why do we have to do that in animals? So first, we can tell the animals that you have to wait for me, otherwise you won't get to marshmallow. They wouldn't care and they would eat the marshmallow. So the big network of many primates can help to design this accumulation and exchange task to test delay of gratification or the primate version of marshmallow test. This field is called I think this one, it just go like when forward when, okay. Oh, how can I, okay. I think there's something weird going on, okay. Or it may be slower than, okay, okay. Okay, oh, okay. Okay, so this field is called comparatory psychology, and it's a network in many primates who try to think hard about which experiment to do, and they distribute this protocol all over the world, so that all the primatologists all over the world conduct the same protocol and combine all the data together, and they try to build these phylogenetic trees of primate cognition. So like we have like at that time we had monthly meeting, but once the project is done, we have less meeting, and then we resume once the new project comes. So they have done already the short-term memory, and the delay of gratification is their second project, and the next one is coming next year. So we try to keep up with them, because the community is so like active, and we learn a lot from them as well. So this is the way to connect to the International Society of the Primatologists. And the main idea of like testing this cognition in primates is that we are thinking that we are not the only primate on Earth. We want to compare ourselves to our neighbors, and we want to understand where we deviate from all the rest. And typically when we put human into that tree that we talk about, human would be like this, and all the others would be like this. I think the most important point is that we have like our newborn, and we kind of grow apart from all the rest of the primates really quickly. After 5-6 years, we are so far away from them, which is really interesting that why we can go so far from them. Okay, and for this project, they, like as I said before, that we think so hard how to test delay of gratification in primates. So the first one is accumulation, because the primatologists believe that delay of gratification or self-control is like the beginning of saving. It's the beginning of exchange when you can wait longer for better reward, or you can give up something in exchange of something else. Because when you buy something, it's going to be really weird that you give me the money first and then I'm going to give you the item. So because of this, our society managed to have exchange. So to do this with monkey, we use accumulation tasks. So we put a bowl in front of a monkey, and then we put a piece of grape one after the other, each one, and then wait another second, and then we keep another one. Once the monkey grab the grape in the bowl, they keep only the grapes they grab. They don't have the rest. And we train them for a while until they know that if they wait until the five of them are in their bowl, they're gonna have all of them. We start with 2 pieces, and then 5, and then in the testing phase, we have 10 items. And then we also increase the time from 2 to 4 to 8 to 12. And then we see which species can wait longer. Yeah. And yeah, we went to the zoo where we have a lot of primates. So Nakhon Ratchasima Zoo is in the door to the northeast of Thailand, which is here. And we were lucky that next to Nakhon Ratchasima there is Khon Kaen, and at Khon Kaen University, there were some students who were interested in primates, so they joined us for the data collection. And at the zoo, there were like 10 species of primates, which is a really rich population of primates. And I think it's the richest in Thailand, because in terms of a public zoo, because I went to the others and they didn't have that many species. So, and there were also a lot of species that our collaborator in the West, they don't have, for example, the gibbons and the langurs. And yeah, these are the 10 species we collected the data from, and in total there were like 36 individuals. Actually, they had more, but one section, which is the macaque section, it was closed. So there were like almost 20 of macaques that we couldn't collect the data, but maybe we can go back for the next data collection phase. So, and for the training, like, as you may imagine, many of them kind of fell off really quickly. But as expected, the apes stay. The chimpanzee and the orangutan stay until the test phase. The macaque, which I think like it was a pity, like it was a shame that we couldn't have like more macaques in our study, because they were like just next to the apes. So with the training phase 1, we just trained them to collect two pieces of grapes, and many of them failed. They just ate the first one and they didn't wait. And then those 4 species waited until 5 grapes, and then only the apes wait until 10 grapes. And until the last test phase, for the 10 grapes, only the orangutan who managed to wait up to the last phase with 12 seconds of waiting. She was really passionate. So the orangutan won. For the exchange, initially we trained them to do the preference test, so to test if they manage to take the bigger reward. So we have like 3 grapes versus 1 grape, and follow if they take only the more reward side. So they pass to the next one. And then the second test training phase, to have the non-food exchange. So we give them an ice cream stick, which is not food, as a currency. And they have to give us the stick back so that they get the grapes. And then once they manage to do the non-food exchange, we're gonna go on with the food exchange. And if we give them 1 grape and wait for them not to eat, and if they don't eat and give us back, we're gonna give them 3 grapes. many of them didn't do that. So, like, yeah, similar to before, but more primates could do the non-food exchange than expected. The gibbons and the langurs and even the little squirrel monkeys, they managed to give us the non-food reward. But up to the food, we could only communicate with the apes. So the chimp and the orangutan like managed to do that. So it's like, it was so clear that, but at the end when we exchanged with more delay, only orangutan won again. But one caveat in this is that the orangutan was trained. He was trained to like ride the bicycle at the zoo, and he knew how to play with like visitors. So that's why he kind of got a bit more passion as well. Versus the chimpanzee, they kind of has less interaction with the passion. So in this game, the orangutan like was the best, but with prior training, so she cheated a little bit. And what's next? So we have done this, and it was like really good experience having worked with like 10 species of primates in a few months, and we had so much fun. But it was only one aspect of self-control. We would like to go deeper in that. So, how are those factors interact in the same individuals? So that's why we want to go on with this. So we created a computerized behavioral task with like 4 different aspects of self-control, like intertemporal choice with the time delay, with the behavioral inhibition, with the accumulation, and effort-based that we want to do in the same animals. And if they manage to learn, we would like to record the neural data to see if they share the same neural basis, neural like circuits. And yeah, last, so I would like to thank the support for this first time comparative psychological experiment in Thailand, self-control studies in Thailand, and allow us to have international collaboration with the primates, many primate networks, and also link to local collaboration, which is the zoos, and they will really welcome us with all the like behavioral tests and the experiment and the behavioral training, because in Thailand, like primate trainers are really like limited, and it's also a welfare issue that they want enrichment for their primates too, because they need a lot of enrichment. And also with university, because we had the opportunity to train the students from the university who were interested in primate behavior. So this was like research collaborations and training, and we would like to go on with this. Thank you so much.
Thank you for a very interesting talk. So now it's open for floor for questions or comments.
Very, very much interesting. So probably I learned, I understand 2 bananas tomorrow is much better than a banana today, but sometimes I need a banana today.
Yes,
so
yes,
how do you explain this situation?
Exactly. So that's like, that's the complexity I talked about with the self-control, because sometimes it's about, sometimes it's about timing, sometimes it's about urgency, because with animals there are so much about passions. Because when the animals is at the lower prey levels, they can't wait for that long, because they're gonna be killed by like bigger animals, or their food will be stolen from someone. So it's also depending on the species as well, and also it depends on the urgency, how hungry you are, right? Sometimes you need to go to the toilet, you can't wait. It's really important. So that's why with those controls, we can approach self-control in many aspects. Some animals may not want to wait, but they are more happy to spend more effort on something else. So I think this series of experiments would be interesting to see if we can manage to do all of them in the same population of animals. Because when we say that self-control is about passions, is about waiting, it's actually not true.
Any other questions?
So from your data, is it possible to take a look at the age of these primates? Does it matter by the age?
Yes, that's a really good question. It's really interesting that I had an occasion to test like informally with the macaques at the zoo, but we didn't have the full data set. It seems like the animals have to become pretty old age before they have self-control. That kind of sparked me a lot. I had a lot of thoughts after that, and I think that if humans we don't have education, maybe we have to grow pretty old before we can self-control. So but like wild animals, they have many challenges, right? So it's harder for them to stay calm. So they have to kind of be born like close to humans so that they can relax and they have more patience and like cooperate, this kind of thing. So, for example, for macaques, they become mature at age 5. They are really like hectic. But until they start to be aged, like maybe 10 years old, it's equivalent to people of like 40 years old. They start to be calm at that age and they can wait longer. And I was like, wow, that's really nice to see. Like I didn't expect that to be that late, but it's really interesting data and I think it's worth like digging into more details. Yeah.
Okay, maybe one quick question. After we know all these behaviors from learning other like non-primates or primates, can we apply to like human? I mean, to train human or maybe educate student, yeah, for more like self-control.
That's my actually ultimate aim of this project. Yeah, I would love to learn more about self-control because it's always amazed me how people like differ in these aspects. We plan also with these four tasks, and if we have some ideas more in the future, we aim to establish a test battery of self-control if they can be indicative of some aspect of self-control, and with the neural signal recording, I think it's going to give us a lot of details about how those aspects of controls operate in the brain. Maybe they use different brain regions for different aspects, and that's going to give us more information about how to tackle those issues. Maybe because of some brain areas have deficit. And of course, yeah, I also have some plan to translate these like tools to, for example, kids, or maybe some patients who have self-control issues, because some neurological disease, they have also self-control issues. And also in some like really challenging areas like addiction and neurological disorders. Yeah.
Okay, thank you so much, Dr. Larita. Okay, so now we come to the end of the seminar. I'd like to thank all of our speakers for their very insightful presentations. So, and also thank you everyone for your participation. Thank you.